Okay, so let us start the fifth unit of industrial microbiology. In this session, we will be discussing the metabolic pathways for the biosynthesis of industrial microbiology products. So, when we talk about the metabolic pathways, uh, understanding the nature of the metabolic pathways is more important in industrial microbiology because understanding the physiology of the organism is necessary to manipulate the organism to produce maximally materials of economic importance at minimal cost. As we know, the objective of the industrial fermentation process is to produce some product of interest, microbial product of interest, which is of some economic value at minimal cost at maximum efficiency. So that requires manipulation of the organism, its production efficiency, its growth conditions, other growth requirements, and even the entire genetic pathway or metabolic pathways of this organism in such a way that you will have your product produced at a maximum efficient and or cheaper levels. Okay. So, for manipulating that metabolic pathway and producing your industrial product in a cheap and efficient way, you require a better understanding of the microbial metabolism for industrial microbiology production. Now, the yeast cell, take the case of an yeast cell. A yeast cell will divide and produce carbon dioxide under aerobic conditions if it is offered with a solution of glucose and ammonium salts. You know that. The increase in the cell number resulting from the growth and bubbling of CO2 are the increase in the cell number. Actually, that is a result of the growth of the organism. And of course, there is a physical observation that can be made, like the effervescence or the bubbling of the CO2 from the medium. Okay, so the increase in the cell number resulting from the growth and bubbling of the CO2 are the only external evidence of the vast number of chemical reactions occurring in that condition, or when the yeast cell utilizes the glucose and the ammonium salts in the medium and grow. The only physical evidence that you can observe is the number of cell or the number of increase or number increase in the cell or the overall increase in the biomass of the cell that can be physically observed. And also there is the evolution of the CO2 gas that can also be observed in during the fermentation process. These two are the external evidences that can be assessed, which indicates the large number of the vast number of chemical reactions that is occurring in the cell. And we don't know what are the different chemical reactions that is occurring specifically. If you want to understand that one, you need a better understanding of the metabolic pathway. Okay. The yeast cell, on absorbing the glucose, has to produce various proteins which will form enzymes and or the enzymes that is required for the catalysis of various pathways and also or of the various reactions, and also for the manufacturing of the different proteins, carbohydrates, lipids, and other components and their breakdown or synthesis, whatever. So, for all the cellular activities, it requires a large number of enzymes, which are basically proteinaceous enzymes, and they are required to be produced in the appropriate or the required quantities. And all these reactions, which is for the growth of the organism, for the synthesis or manufacture of the proteins and utilization of the carbohydrates or lipids and other cellular components. All the chemical reactions that is occurring in the cell is actually guided by different enzymes. All these required enzymes needs to be produced in the system with a specific pathway. Now, the series of chemical reactions involved in converting a chemical or a metabolite you say, especially in the cellular context, we will say all the chemicals were basically metabolites. Okay, so converting a chemical or a metabolite in the organism into a final product is known as a metabolic pathway. So there is a series of chemical reactions which is required for converting one chemical or a one metabolite into a final product, and that pathway or the series of chemical reactions is known as the metabolic pathway. And we know that there are basically two types of metabolic pathways, which is categorized as anabolism or anabolic pathway, and the catabolism or the catabolic pathway. And there we have during all both these pathways or in between these pathways we have various intermediates. So because when we talk about a pathway, it is not a single step conversion, it involves various steps. So means A will be converted to B, B will be subsequently converted to C and D. So in the conversion of A to the final product D, which is known as the end product, there are various intermediates like B, C, etc. So in every metabolic pathway, we have a metabolite or a chemical that is required to be converted into the final product or the end product, and there are various intermediates that is available. So the economic importance or the industrial importance may reside in sometimes in the end product, or sometimes it may be the intermediate that you are looking for as the final product. Okay. The nature of when we in the nature of the metabolic pathways, the first category that we want to discuss is about the catabolic reactions. So the catabolic reactions have been mostly studied with glucose. It's a standard method for studying such metabolic pathways. So these catabolic reactions often furnish energy in the form of ATP and other high energy compounds which are used for biosynthetic reactions. So basically catabolic reactions were breakdown reactions or utilization reactions where some metabolite will be degraded or some chemical will be degraded, and finally it will yield energy and other ATP like energy like ATP or other energy rich compounds will be generated, and that energy can be used for some other biosynthetic reactions. So the cell requires various forms of energy like ATP, GTP, or etc., etc., and that will be utilized in various metabolic pathways like biosynthetic reactions for synthesis of the amino acids, etc. Okay. The second function of the catabolic reaction is to provide the carbon skeleton for the biosynthesis. So when the cells grow, divide and grow, it requires a synthesis of a carbon skeleton, or the cell mass or the biomass needs to be increased during that growth. So that needs to be synthesized during these catabolic reactions. That anabolic reactions lead to the formation of large molecules, some of which are constituents of cells like proteins. Anabolic reactions were basically synthetic reactions or biosynthetic reactions which produce some complex or new compounds, larger molecules like the proteins which is produced from the amino acid. The amino acid biosynthesis and subsequent production of the proteins can be regarded as an example of an anabolic reaction. Now, this picture shows the catabolism and anabolism, the pathways. In this case, in the case of the catabolism, you will start from the top and move down. Like these are energy yielding metabolism, and here we have an energy source or a metabolite that needs to be degraded. So on breaking down of this energy source or the metabolite, that will lead to the production of various metabolic products and also release of the energy. That energy is utilizable, and that basically when it is released, it will be transferred to the ADP, and ATP is synthesized. So the ATP is the energy currency in the system which can be supplied to the various biosynthetic pathways in the form of energy. So that's how the energy yielding mechanism of the catabolism works, where a high energy compound will be broken down into simpler products along with the release of the energy which will be used or transferred to the high energy, the energy carriers like ATP, which will be transferred subsequently to the utilization sites like anabolic reactions or the biosynthetic reactions. Now, the second picture needs to be read from the bottom to top, where we have the synthetic approach or biosynthetic approach is adopted. Here, the external nutrients or basic nutrients will be utilized in the cell for synthesis of a complex compound or a complex structure. There we will have intracellular precursor pool, and the ATP or the energy supplied through the ATP is utilized in the biosynthetic reaction, and some biosynthetic intermediates like amino acids were produced, and using that amino acids, biopolymers like proteins or complex proteins will be produced. So this is an example of an anabolic reaction in which energy is utilized along with some similar nutritional elements and other precursors, which will be utilized for the production of biosynthetic intermediates like amino acids and the complex compounds or biopolymers like proteins. So these are the two different catabolic and anabolic reactions that is occurring in a biological system. Am I clear and audible? Okay, fine. Now, some pathways have elements of both kinds of and metabolic, both means there we have metabolic intermediates which are derived from catabolism and it is utilized in anabolism. For example, say compound A is broken down to B and subsequently converted to C. So the B is an intermediate of the catabolic reaction now. But when the B is utilized for synthesis of say X and then subsequently converted into Y. So there is an anabolic reaction which utilizes this intermediate B as a building block. In that case, for the second pathway of the anabolic pathway, the B is a building block which can be subsequently converted into a new compound like another protein or another complex structure. So if an anabolic pathway utilizes an intermediate that is created during the catabolic process, then the intermediate actually represents or it is involved in two pathways. One, a catabolic pathway, so it is produced during the breakdown of a compound. Then subsequently that is utilized again in an anabolic pathway for the synthesis of something. So that intermediate which is coming in both the cases like catabolic and anabolic pathways, there this type of intermediates were known as amphibolic intermediates. So it is actually coming in between two pathways in the anabolic pathway and or it is coming as an intermediate of an anabolic pathway and a catabolic pathway. So that can be taken for example, the intermediate of the catabolic pathway can be taken as a building block for, or intermediate block for the anabolic pathway. So in that case, it is known as a amphibolic intermediate. So the type of compounds were also present. Now, when we come to the different products of industrial microorganisms, the products of industrial microorganisms were broadly divided into two groups. And they are primary metabolic products of primary metabolism and products of secondary metabolism. This graph shows the primary fermentation and secondary fermentation. And the primary fermentation, we know that the graph actually it shows the cell number increase on the y-axis and the time on the x-axis, where usually primary fermentation follows the same pattern or the production of the ethanol, for example, which is a primary metabolite. It follows the same pattern as that of the cell or biomass increase because as you know in the normal growth curve in a system we'll have a lag phase and a log phase and a stationary phase, then subsequently a decline phase, and this production of the primary metabolite follows the same pattern. So it almost follows the same pace of production as that of the biomass. Whereas in the case of secondary fermentation, we will have a separate mode of production where the cell will follow the normal growth curve pattern where we have a log phase, lag phase and a then stationary phase and decline phase, etc. Whereas the secondary metabolites, like penicillin, it will start its production only, say, at the stationary phase or at the late log phase. So it starts production at a later stage in the late log phase, then starts or it reaches its maximum when the decline phase started. Okay, after the stationary phase or when there is some decline in the or depletion in the nutrients occurs, then it reaches the maximum production. So in the case of this graph, you can see that it is divided into trophophase and idiophase. The trophophase almost covers the log phase and lag phase, or lag phase and log phase, whereas the idiophase, which covers the stationary and subsequent decline phases. And the secondary metabolite production basically occurs in the idiophase. Okay. So this is another picture in which we have the biomass, mass of yes, biomass and substrate, etc. Or the product is represented here, and this is the time. In this case, during the growth, you can see that in the case of primary metabolite, the substrate will decline. Naturally, it is utilized and decline. And in the primary metabolite and the product, primary product and the cell biomass production will almost keep the same pace, and the substrate is decline. Whereas in the secondary metabolite production, the substrate will decline, the cell biomass increase it follows the normal pattern, and in the idio phase or the late phase where substrate is depleted, and the cell, after the stationary phase when there is a restriction in the growth condition, and at that stage the secondary metabolite productions reaches its maximal level. So this is the production stages of the primary and secondary metabolites. So if you are designing a pathway or an industrial process, you should look at the phase of growth of the organism. So if you are looking for a secondary metabolite, you have to ensure that the growth phase of the cell reaches that particular idio phase for a secondary metabolite or maximum production of the secondary metabolite. If you are continuously supplying substrates and keeping it continuously in log phase in a culture and looking for maximum production of a secondary metabolite, you will never achieve it because the cell biomass will increase, and during the top phase, you can't expect a secondary metabolite production or a maximal production of the secondary metabolite because we need to transfer the cell or the growth stage into the idio phase, not to the trophophase. Okay, so based on the product requirement, say if it is a primary metabolite like ethanol, you have to ensure that your growth phase is remaining in the trophophase. Or if you are looking for a penicillin or some other secondary metabolite, you have to design the growth conditions in such a way that it is maintained at the idio phase. Then only you can achieve the maximum production. So this is the concept. Okay. Now, the primary metabolism is the interrelated group of reactions within a microorganism which are associated with growth and maintenance of the cell. So the key term is the growth and maintenance of the cell. The role of the primary metabolites is the growth and maintenance of the cell. Primary metabolism is essentially the same in all living things and is concerned with the release of energy and the synthesis of important macromolecules such as proteins, nucleic acids, and other cell constituents. When primary metabolism is stopped, the organism dies because the growth and maintenance of the cell is absolutely necessary for the organism to live. So if there is some problem in that metabolism, the organism normally dies. So these metabolites, primary metabolites, are usually associated with the growth, and maximum production of these primary metabolites occur in the log phase or the trophophase of the growth in a batch culture, as we discussed earlier in the earlier slide. The primary catabolite production include catabolic products include ethanol, lactic acid, butanol, etc. So these are catabolic products of primary fermentation or primary metabolism: ethanol, lactic acid, and butanol. Whereas primary anabolic products include amino acids and nucleic acids. So these are some of the primary products which are absolutely required for the existence of the cell or growth and survival of the cell. Single cell proteins and yeast would also be regarded as a primary product. So single cell proteins were basically biomass, and that can also be treated. And you know that biomass will reach its maximum production in the log phase. So these single cell proteins and yeast, etc., can be considered as a say primary products of metabolism. This table shows the different anabolic and catabolic products of primary metabolism. In the first list, we can see the anabolic products, including the enzymes, amino acids, vitamins, polysaccharides, then yeast cells, single cell proteins, then nucleic acid, citric acid, etc. These are anabolic products or synthetic products. So it is a product of biosynthesis of primary metabolism. Whereas the catabolic products or the breakdown products of the primary metabolism is also there, like ethanol and then butanol, acetone, lactic acid, then vinegar or acetic acid, etc., are examples of primary catabolic products. So these are some of the industrially important primary metabolism products in the anabolic category and catabolic category. Now, in contrast to the primary metabolism, secondary metabolism has the following characteristics. First of all, we will list the different characteristics. We have, say, 7 characteristics that is listed. First one is there is no apparent function in the organism. Then it is produced in response to restriction in the nutrients. Then it then restricted to some species of plants and microorganisms. Then it's having some bizarre or unnatural structure. Then it is subjected to strain degradation or strain degeneration. Then it is also stated that it is controlled by plasmids. Then it is also triggered by the inducers for its production, secondary metabolism products. So we will see it one by one. So we have 7 points to be discussed at this point of the secondary metabolic products. And the first point is about there is no apparent function in the organism. In the previous case of the primary metabolites, we had discussed that it is absolutely required, and if it is not produced, the organism will die. But in the case of a secondary metabolism, we can say that there is no apparent function in the organism. So if it is not produced, the organism doesn't have any difficulty at all in growing. Okay. The organism continues to exist if the secondary metabolism is blocked by a suitable biochemical means. On the other hand, it would die if primary metabolism is stopped, as we discussed. Then these are produced, or secondary metabolites are produced in response to restriction in nutrients. That's also discussed in the previous slides, because when the nutrients were depleted and when it reaches the decline stage or late idiophase of the growth, then the secondary metabolites will reach its maximum production. So it is produced in response to restriction in nutrients. They are therefore produced after the growth phase, at the end of the logarithmic phase of the growth, and in the stationary phase and decline phase in a batch culture. So they can be, so in that phase can be better described as the end of the logarithmic phase, and the stationary phase can be better described as the idiophase of growth. Okay, they can be more precisely controlled in a continuous culture. Okay. So the next point is about secondary metabolism appears to be restricted to some species of plants and microbes, for example. Of course, there are some cases of some reports about the animals also, but basically the secondary metabolites were obtained from plants and microorganisms. The products of secondary metabolism also appear to be characteristic to a species. Okay. Both of these observations could, however, be due to the inadequacy of the current methods of recognizing the secondary metabolites. That needs to be mentioned, because we can make a general statement that we are nowadays obtaining secondary metabolites from plants and microorganisms. So it's a general statement based on our current observations. But for identifying a secondary metabolite, we require specific tools like separation tools and specific chemical structure determination tools, etc. That is required. So if you can identify a secondary metabolite, or if you cannot differentiate between two secondary metabolites or a modified form of a secondary metabolite, then you cannot identify it. Okay. So if you want to identify a secondary metabolite in an animal, first of all, you have to you require specific tools for identifying that. So it doesn't mean that there are secondary metabolites available in animals or some other species, but our current methods were not sufficient for identifying all the possible secondary metabolites and at the structural level. Okay. So anyway, basically nowadays the studies on secondary metabolites were basically focused on plants and microorganisms. Okay. The secondary metabolites usually have bizarre and unusual chemical structures, and several closely related metabolites have been, or it may be produced by the same organism in the wild type strains. So the bizarre or unusual structures of the secondary metabolites were basically uh, produced in different strains of the same organism. For example, wild type and another type. So the two types of strains will produce a closely related, maybe with one or two groups will be added to the chemical structure. So it will become a different compound. Just like, for example, we are considering the antibiotic or something else, then the antibiotic will change its entire nature and resistance property if it is attached with another group, right? So similarly, if such modifications were occurring, chemical modifications were occurring in one strain and one strain to another, then it will create a lot of unusual chemical structures or bizarre chemical structures can be expected in the case of secondary metabolites. This later observation indicates the existence of a variety of alternate and closely related pathways. So in every organism, there are a lot of alternative chemical pathways will be there, which will be used for modifying the base structure of the compound. So in one strain, it will be subjected to one pathway. It will add some groups, and you will get one structure. And if another strain of the organism or another organism is adopting a separate pathway for adding the groups and modifying the structure, then you will get another compound. So secondary metabolites and its various derivatives were observed in the case of various organisms and its wild type strains. So the problem is that we can't differentiate between such type of closely related or structurally related compounds. We don't have the scientific tools or instruments for separating these two closely related compounds and identifying what are the different functional groups which is attached or the groups that is attached to that chemical structure. So that's one of the limitations in identifying the related metabolites or secondary structures. Now, the ability to produce a particular secondary metabolite, especially in industrially important strains, is easily lost, and this phenomenon is known as strain degeneration. So, you can have a strain with a particular ability to produce a secondary metabolite, and after some times of subculturing, you will see that the strain lost its ability to produce that particular metabolite. And that is due to the loss of these genes that is required or control elements that is required for the production. So it is known as a strain degeneration. So if a strain, industrially important strain, loses its ability to produce a secondary metabolite, then that is known as a strain degeneration. So such type of secondary metabolite producing strains were subjected to strain degeneration. Owing to the ease of the loss of the ability to synthesize secondary metabolites, or due to the problem of strain degeneration, particularly when treated with some mutagenic agents like acridine dyes or exposure to high temperature or other treatments, which may induce plasmid loss, the secondary metabolite production is may be lost, and it is believed that it is because they are controlled by plasmids rather than the organisms, say chromosome. So if it's a chromosomally controlled gene, then it may not be that much affected by such mutagenic agents like mutagenic agents or such a high temperature or other treatments, mutation treatments will not be effective. But if it is a plasmid controlled gene, it will be easily affected and it will be subjected to strain degeneration. So, based on some of the observations in which you can induce a strain degeneration by subjecting it to some mutagenic conditions, then we can argue that, or we can reach to a conclusion that most of the metabolite production were controlled by plasmids. It doesn't mean that all the genes that is required for metabolite production or the pathway is residing in the plasmid. There are a lot of cellular genes and that enzymes will be used in the metabolic pathway, but it is controlled by the plasmid. So the controlling elements for that secondary metabolite production may be present in the plasmid. So if the control is lost, then the other chromosomal genes which is involved in the same pathway may not be effective. Okay. So basically, the strain degeneration is occurring because it is the production of the metabolite is actually controlled by the plasmids. That's why. So the factors which trigger secondary metabolism is also known as inducers. So secondary metabolites basically for its production it requires some inducer molecules, and these inducers is also required for morphological changes and chemical changes which is occurring in the organism. Both is required for metabolite production. So the morphological changes and the concerned chemical changes. The chemical changes will result in the production of the metabolites, and the morphological changes will create some visible structures or visible growth patterns in the concerned organism. So the inducers will result in the production of morphological changes and chemical changes. The chemical changes were actually the production of the metabolite. So all the required genes will be activated and the metabolite will be produced. That's the chemical change. Whereas the morphological changes were actually a distinguishable growth pattern that is shown by the concerned organism. We will see an example. Is it okay? Clear? Hello? Okay. Now, there are some inducers, and they are known as auto-inducers. Auto-inducers can be, some examples can be listed as the gamma-butyrolactones or butanolides of as actinomyces. So in actinomyces, we have gamma-butyrolactones. And in gram-negative bacteria, we have HSLs, that is N-acyl homoserine lactones. Then in gram-positive bacteria, we have some oligopeptides. Then amylolactopsis mediterrane, there is another organism where we have a B factor that is involved in the production of rifamycin production. So we have various types of autoinducers: gamma-butyrolactones for actinomyces, N-acyl homoserine lactones for gram-negative, oligopeptides for gram-positive, and B factor for amylolactopsis mediterrane. So these are some of the examples of autoinducers. And now we will see some of the details about some of the autoinducers, and they function in the development. Actually, these autoinducers will function in the development, sporulation, light emission, virulence, and production of antibiotics, pigments, cyanides, plasmids. So various results will be there for the autoinducers. So it will produce some metabolite or secondary metabolite, and the resultant morphological change in the organism may be different, like sporulation, light emission, or fluorescence, then virulence of the organism, or production of an antibiotic or pigment, etc. So the morphological change will be varying in different conditions, but all these type of changes actually it requires some type of auto-inducers. And of great importance is the actinomycete fermentation with gamma-butyrolactone, the first one that we discussed. And here we have an A factor. The previous slide, once again, the B factor is another one. The gamma-butyrolactone is the first category that we are going to discuss. The A factor is a type of gamma-butyrolactone. A factor is a 2S isocaprylyl 3R hydroxymethyl gamma-butyrolactone. That's the full name of that A factor. So A factor is one example of a gamma-butyrolactone of actinomyces. The A factor, it induces both morphological and chemical differentiation in Streptomyces griseus and Streptomyces bikinensis. So we have two Streptomyces species, and the A factor is an inducer coming under the gamma butyrolactone category, and it is having two functions, that is a morphological change and morphological and chemical differentiation. Both will be done by the A factor. So, what are the different types of morphological changes? Aerial mycelia and formation of conidia. So, these are growth pattern or growth changes in the organism, morphological changes of the organism. Then, what are the chemical changes? Production of streptomycin synthetase enzyme and streptomycin. Streptomycin synthase enzyme and streptomycin production were chemical changes or chemical differentiation of the concerned organism. So both will be resulted by the inducer A factor. So morphological and chemical differentiation of Streptomyces griseus and Streptomyces bikinensis is occurring, and that results in the production of aerial mycelia, conidia, Streptomycin synthase enzyme and streptomycin. Okay. Conidia can actually form on agar without A factor, but area mycelia cannot. So on agar plate, if you are growing the Streptomyces griseus without A factor, it will grow only on the surface. The aerial hyphae or aerial mycelia, it cannot rise from the agar surface. It will stick on to the, or it will agar surface and it will grow. So there are mycelia or conidia etc. can be observed, but it will not rise from the agar surface. So if A factor is there, then it will form aerial mycelia and conidia will rise above the agar surface, and you will have an aerial growth of the concerned actinomycete. The spore form of the spore actually it forms on branches. Actually, it forms on the branches, and morphologically it is similar to the aerial hyphae. But the only difference is that it will not emerge from the colony surface. So only in the presence of the A factor, the hyphae, aerial hyphae, will emerge from the colony surface above the agar plate. In Streptomyces griseus, A factor is produced just prior to streptomycin production, and it disappears before the streptomycin is reaching its maximal level. So it induces at least 10 different proteins. One among them is streptomycin 6 phosphotransferase. So at least 10 different proteins, including streptomycin 6 phosphotransferase, is produced by this A factor. So it triggers the transcription. At transcription level, it induces all this the production of these 10 proteins, and which will function in the morphological and chemical differentiation of the concerned organism. In A factor deficient mutant, there is a failure in the transcription of the entire gene cluster, so none of the genes will be coded, so resulting in the lack of this aerial hyphae production and streptomycin synthetase production. Many other actinomyces produce A factor or related gamma butyrolactones, which differ in the length of the side chain. So there are other actinomyces which may produce A factor, but their role will be different. Maybe the gamma butyrolactone will have a structural change like length in the side chain etc. will be there. That will have some other function in some other organism. So similar gamma butyrolactones were present in many organisms, many type of other than the Streptomyces griseus and bikinensis. We have various other organisms which may produce other types of antibiotics. So in that case, they will have their own role. Such respective gamma butyrolactones will again result in their morphological differentiation and production of the concerned antibiotic. So such type of different inducers will be there, but the specifically speaking, the A factor is in this case is specifically used for these particular organisms. That is Streptomyces griseus and Streptomyces bikinensis, not for other ones. And for other actinomyces, there may be other gamma butyrolactone with their own combination of morphological changes, and different set of enzymes will be or proteins will be produced by the concerned cassette or transcriptional cassettes. Okay. So these are some of the list of secondary metabolites produced by the microorganisms. We have antibiotics, pigments, toxins. then effectors of ecological competition and symbiosis, then pheromones, enzyme inhibitors, immunomodulating agents, receptor antagonists and agonists, pesticides, then anti-tumor agents, growth promoters of animals and plants, alkaloids, then a wide variety of drugs and toxins, then other useful materials like gibberellic acid and plant growth hormones, etc. So we have a lot of secondary metabolites nowadays produced, and that is why having wide applications. So this is another table that shows the different type of secondary metabolites. The first category is antibiotic. So antibiotics, typical examples include penicillin, streptomycin, etc. So it is having clinical use, as you know. Now there is anti-tumor agents like actinomycin, bleomycin, etc., which is again having this clinical use. Then toxins like aflatoxins, amanitin, etc. Then alkaloids, which is having pharmaceutical use. Ergot is an example. Then there are other miscellaneous compounds like gibberellic acid, kojic acid, muscarine, etc. Okay. They have a major effect on the health, nutrition, and economics of our society, the secondary metabolites. They have unusual structures, as we discussed, bizarre and unusual structures are there. And their formation is regulated by the nutrients, growth rate, feedback control, enzyme inactivation, and induction, etc. We will have a discussion on that topic in subsequent lectures. Their regulation is influenced by unique low molecular mass compounds like transfer RNA, sigma factors, and gene products formed during the post-exponential development, etc. So their regulation and other things will be discussed as I mentioned. It will be discussed in the subsequent topics. The synthesis of these secondary metabolism are often coded by clustered genes on the chromosome and infrequently on the plasmids. So basically, the synthesis of the secondary metabolites were present in the chromosome itself, whereas it's a regulatory elements where, as we have discussed in one of the previous slide, it may be present in a plasmid or it is controlled. The control elements may be present in the plasmid. That's why it is actually easily subjected to strain degeneration. Whereas the other genes or the cassette of the genes that is required for the secondary metabolite production and the synthesis and other enzymes were present in the chromosomal DNA itself. Unlike primary metabolism, the pathway for secondary metabolism is still not understood to a great extent. So the primary metabolism is it's easy to study because it affects the cell growth easily, and we can manipulate them and easily identify, and that process is very easy. But since the secondary metabolite pathway is a complex pathway and it is not actually expressed in the cell as such because it is not required, So in that case, to study the pathway of a secondary metabolism is difficult when you are comparing it with the primary metabolism pathway. So also it is a little bit more complex when we compare it with the primary metabolism pathway. So our methods and means for understanding the secondary metabolism pathway is a little bit difficult. So in that case, as you know, we require more sophisticated tools and techniques for understanding the complex secondary metabolism pathways. Secondary metabolism is brought on by, as we discussed, it is brought on, or the metabolism is actually promoted by exhaustion of the nutrients or substrate depletion, and also by biosynthesis or addition of an inducer. So if you require the secondary metabolite, or if you want to increase the secondary metabolite production, you have to ensure that it is depleted with the nutrient, and also you are providing it with an inducer. And also there should be a decrease in the growth rate. So if the organism remains in the log phase of growth by continuously increasing biomass, then you can't expect the secondary metabolite production at its maximal efficiency. And these events generate signals which affect the cascade of regulatory events resulting in chemical differentiation and morphological differentiation. So the previously stated three events like exhaustion or depletion of the nutrient, addition of the inducers and also decrease in the growth rate. So that means these conditions will stimulate the regulatory cascade, resulting in chemical differentiation and morphological differentiation. The chemical differentiation means the expression of the entire cassette or the set of the genes that is required for morphological differentiation and the concerned enzymes that is required for the secondary metabolite production. And the morphological differentiation is the changes of the growth, like production of the aerial hyphae, etc., etc. So that will be resulting from the secondary metabolite gene activation. The signal is often a low molecular weight inducer which acts by negative control. So that's by binding to and inactivating a regulatory protein. So usually the inducer will bind to and inactivate a regulatory protein, which normally prevents the secondary metabolism and morphogenesis. So you might have learned that lac of proton concept, in which the inducer molecule will relieve the repression. You know that one. So similarly, here in the case of an inducer molecule, it will relieve the repression of the concerned genes, regulatory genes that is involved in the secondary metabolism and morphogenesis of the organism. Thousands of secondary metabolites are widely different chemical groups and physiological effects on humans have been found. Okay, so there are already reported thousands of metabolites are there, and the vast potential utility of these microbial secondary metabolites is yet to be realized. They can be and many of these metabolites are yet to be discovered, because we have or we require more efficient tools for the identification of such secondary metabolites, and also we require specific applications. For example, whether it is an antimicrobial compound or an anti-cancer compound or some other function for that particular metabolite needs to be studied in detail. So in that case, we require more advanced techniques for identification of such complex structures and studying their biological or biological applications. So this is about the general concepts about the primary and secondary metabolites. Any questions? Hello? Okay. So let me stop.